Good afternoon. My name is Don DeMeo, and I'm an associate professor at Harvard Medical School and an associate physician at Brigham and Women's Hospital in Boston. It is my pleasure today to speak to you about sex and the aging lung, chronic obstructive pulmonary disease. The objectives of this talk are to include an overview of associations with sex and gender in lung disease, and note a role for both the genome and the epigenome in sex differences in the lung. I will propose a role for networks and network medicine as a way to integrate sex and gender differences for the understanding of COPD. And last, I hope by the end, you'll be convinced that the path to precision medicine for pulmonary diagnostics and therapeutics is through the science that captures sex and gender-related causes and effects in a holistic fashion. Now, COPD, or chronic obstructive pulmonary disease, is a disease state characterized by airflow obstruction that is not fully reversible. And what we see here on this slide is two graphs. The top left shows age on the x-axis and forced expiratory volume on the y-axis, and that is a common measure of lung function. And what we see in the red line is the suggestion that some individuals have accelerated lung function decline across the life course. If we look at the lower right slide, what we see across that life course is that the normal trajectory of lung function decline includes the development of what's called senile emphysema, a normal aging process. But the suggestion of the development of telomere shortening, cellular senescence, and other hallmarks of aging puts COPD in the category of diseases associated with age acceleration. As we trek towards precision medicine in the lung and other complex diseases, we think about the compendium of risk factors that accumulate across the life course, starting with in utero development, proceeding through the game of life to adolescence, and then finally in advanced age with lung dysfunction and symptomatology. And if we think in terms of sex and gender, here I present the National Vital Statistics from 2017, males on the left, females on the right, which show that although chronic lower respiratory disease, of which COPD is a part, was the fourth leading cause of death in men, it was the third leading cause of death in women. And now in 2021, we know that COVID has supplanted the COPD rankings, but it is still a major cause of death in both men and women. Here I present an infographic from a paper we published in Lancet last year, which separates out biologic and gender-related differences. So sex, as the biologic construct, includes those issues related to sex chromosome complement and may impact lung development, disease pathophysiology, and this is in contradistinction to gender-related issues, which capture lifestyle, sociocultural aspects, and may include issues related to smoking. We know that lung disease is one of the diseases that globally has been associated with health disparities. And what we also know is that sex differences have been recognized as one of the most significantly understudied aspects of human health research. Many prior experimental studies and trials do not include equal numbers of males and females, and sex chromosomes have generally been excluded from genomic analyses. In 2015, NIH guidelines suggested that we all need to consider sex in research design analyses and reporting of scientific findings, and it is only through the accounting for biologic sex differences can we improve our management of disease prevention and therapeutics. Now, no discussion of COPD is complete without a discussion of smoking and lung health. This is a beautiful paper by Don Sin and his group. It's a little bit on the older side, but it really captures the data that persists today, and it summarized the data regarding whether women versus men have increased susceptibility to the effects of tobacco smoke. And this was a very comprehensive review. And what it noted was that beyond age 45-50, female current smokers had significantly faster annual lung function decline than male smokers. And this became very important in the year 2000. And this was the first time that the number of women dying from COPD exceeded the number of men. And this projected increase in disease morbidity and mortality has continued through today. And what we know, when these trends and patterns now were repeatedly studied, and this is a paper published in JAMA in 2017, what we saw was an overall continued increase in respiratory mortality between 1980 and 2014, with the highest mortality in the central Appalachian Valley of the United States. Although COPD mortality rates in this study were noted to decrease, nationally, COPD mortality rates in women increased more than 100%. And when we look at the differences between men and women with COPD, we know that women tend to be underdiagnosed and suboptimally treated, that they may differ in their disease presentation, that tobacco use susceptibility varies, and that the relationship between the development of disease and other exposures, such as occupation and non-occupational exposures, may portend differential risk between males and females. We studied gender-related differences in our lab group, and this is work by Ed Silverman, where he noted that the age of onset considerations may lead to novel insights for understanding the pathogenesis related to sex, age, and race differences. And these are data from the Boston Early Onset COPD study. And across all phases of enrollment of this very unique cohort, which included advanced lung disease before age 53, the majority of participants were females. We followed this up. This is research performed by Marilyn Foreman, who was a postdoctoral fellow at the time. And what she noted is if you look across the age groups in the COPD gene study, with age on the x-axis and the percent of the cohort on the y-axis, and you look across the age bins, at the youngest age group, more women had advanced lung disease than men. So once again, reinforcing increased prevalence of early severe disease in younger women. So we reflected on these repeated findings and asked, why this female predominance of early onset COPD? Well, it could be because women have smaller lungs than men. There may be shorter survival in men with COPD. COPD could be an X-linked dominant disease, but that doesn't seem to be the case. There could be hormonal issues, differences in tobacco smoking related to underreporting, but none of these seem to be the case. And we were really honing in on this idea that the increased female susceptibility may be by gene by sex and gender interactions and a complex interaction with aging. We know that if we look at studies that have focused on the menopausal transition, that lung function declines more rapidly at the menopausal transition, and that we urge clinicians to be aware that respiratory health deteriorates during reproductive aging. We performed a study, again in the COPD gene study, where we looked at differences parsed by age less than or greater than age 65. And in these groups, we saw that women tend to have increased risk for shortness of breath, that's dyspnea, exacerbations or periodic worsening of their lung function, and more severe COPD. And again, it pointed us towards accumulative increasing severity with aging. So as I noted, we were honing in on this complicated interaction between the genome, sex, and advancing age. This paper is a beautiful summary of the role of sex in genomics of human complex traits, and I urge everyone to read it. Sex-specific genetic architecture has been described for a host of human traits, including lung function, and failing to model the sex-specific architecture will really hamper the detection of susceptibility loci for complex disease. Up until recently, ignoring the X and Y chromosomal variants has been standard, but we really miss a whole host of regulatory genomic interactions that may help us better understand the differential risk for lung disease in men versus women. Again, thinking about the state of the literature in the mid-2000s, there have been several integrated studies of sex-specific and gender-specific manifestations of COPD, and this data by Fernando Martinez and his group analyzed CAT scans, which are advanced X-rays, and looked at quality of life data from a clinical trial. And they noted that women with COPD had more shortness of breath, just as we had noted recently, and had lower reported health status. but they also demonstrated that women had less emphysema, which is a very destructive phenotype. And when they look at the histology of the lung, women had thicker airways. So this idea started to be suggested that maybe emphysema is a phenotype of men. So we investigated this and looked at sex-specific features of emphysema in the COPD gene study. And if you focus on the histograms on the right, when we looked at all subjects, it was true that men had on average more emphysema. But when we looked at in subgroups with early onset disease, severe emphysema, or advanced lung disease, there was no difference. So emphysema is not a phenotype of men. And we looked at the genetics of this and described a sex-specific association with emphysema in women in the SELSR1 gene, but not in men. And this SELSR1 gene is quite interesting because it had previously been linked to normal lung branching morphogenesis, which is a key developmental stage in the lung. Again, we were heading into this idea not only about aging, but maybe sex-specific aspects of lung development may portend differential risk for disease. And as we were advancing this idea of the investigation of the genetics by sex for COPD, I started thinking about this idea of the evolution of sex differences in human disease. And I think this paper by Morrow and colleagues says it best. We don't really expect that the general phenomenon of sex differences to be related to the genetic alphabet or genetic variation, because the genetic material is shared, but maybe it is related to the epigenome. And the epigenome, broadly defined, are elements that may change the expression of the gene without changing DNA sequence. And we started pursuing the investigation of DNA methylation, which is one example of an epigenetic variation. We looked at smoking-associated site-specific differential methylation in cheek cells in the buccal mucosa, and we compared male and female results for differential methylation at the CYP1B1 gene. And CYP1B1 is interesting because it is a xenobiotic metabolizer of cigarette smoke. And what we observed was that differential methylation at this gene was associated with lower lung function, this is FEV1% predicted, lower ratio of FEV1 to forced vital capacity, as well as percent emphysema in women, not men. And we followed it up looking at lung tissue. And what we observed when we looked across the genome with chromosome on the x-axis and the negative log of a p-value for a genome-wide DNA methylation evaluation, there were a lot of differential methylation marks in lung tissue that varied by sex. And when we modeled a sex by smoking interaction, we saw several marks had different directions of effect between males and females. Again, bringing us to this idea that maybe it's the regulatory genome that's important for sex differences in COPD. We've also looked at the metabolome, and this is a paper that was recently published looking at metabolomic differences between men and women with and without COPD. And we identified a whole host of metabolites that were different between men and women. So when we think in terms of the canonical pathways that take us from DNA variation to proteomic expression, we think in terms of layers of the genome. And when we think in terms of layers of the genome, we think of a network. And we're very interested in network and systems biology approaches brought to bear in understanding lung disease. So what is a network? What is network medicine? Network medicine broadly represents, as defined in this paper by Laszlo Barabasi, is the study of cellular, disease, and social networks which aims to quantify the complex interactions contributing to individual disease. And a real unique aspect about network medicine is that it's holistic rather than reductionistic. It allows us to take the compendium of molecular disease layers and construct a comprehensive model. It may allow us to capture nonlinear effects and emergent properties, and it broadly employs systems biology methods. And we've thought about this in several different ways as we thought about the sex and gender findings related to causes of death, as we thought about gender differences and sex differences in cancer, the differential methylation, and we emerged upon this idea that we should look at sex differences in gene expression and regulatory networks first in normal tissue. And we had started thinking about this in 2014, and this is a paper by Kimberly Glass in our group. And what we did was we built gene regulatory networks in men and women with COPD. And what we broadly observed is that whether we looked in sputum, so hopefully representing the lung, or if we looked in blood, we could build gene regulatory networks that identified different nodes and edge connections for females in pink and males in blue. We scaled this up to the GTEx consortium data, and we built 8,279 single networks. We first looked at differential gene expression in these samples across 29 tissues. And on average, except for the sex chromosomes, we did not see broad differences in sex-specific gene expression across all the tissues. However, when we built gene regulatory networks, we saw that although on average there was no differential gene expression, there was a big difference in how genes were targeted between men and women. And so the way I like to phrase this is that the genes end up in the same location, but the road they took to get there seemed to be different between men and women. So a gene regulatory network is one way to try to understand sex differences in lung disease. We also looked at co-expression networks, and we built co-methylation networks looking at correlation between methylation, and we first started to do this in lung tissue. Again, going back to the aging hypothesis, we built lung tissue correlation networks from DNA methylation data in both fetal lung tissue, fetal lung DNA methylation, and adult lung tissue from adults with chronic obstructive pulmonary disease. And what we revealed is that there was a lot of similarity in the networks between the fetal lung in early life and COPD in later life, focused on Wnt, Hippo, and other pathways that have been related to aging as well as lung disease. And this is a paper published by one of my postdoctoral fellows, Priya Kachru, and what we saw was that DNA methylation perturbations may link development and aging in the lung. And bringing this all together, we've also observed these findings for sex differences as well. So bringing it all together, we really start to see across the layers of the genome, across the different genomic signatures we can develop, starting with DNA variation, DNA methylation, gene expression, metabolomics, proteomics, and then a holistic network, we start to see evidence that COPD is a disease of aging, it's a disease of sex differences, it's a disease of sex differences that may start in utero and progress across the life course, and it's a disease that is differentially impacted by environmental exposures such as cigarette smoke between men and women across the life course. So why is this interesting? Why is this important? It really brings us to clinical considerations. I'm a pulmonologist, and we know that women carry the excess burden from COPD in terms of being younger with advanced disease. They have more shortness of breath for similar levels of lung function impairment, increased healthcare utilization, and this has therapeutic implications. We know that women may have increased susceptibility to tobacco smoke, and I should say that both men and women should stop smoking. It's never too late. But we know that smoking cessation may bring about an even greater improvement in lung function in women. Aging in the lung with differences by sex may drive the differences we see in COPD, and we really need to give consideration of modeling the molecular targets, drug responses, the networks with sex and gender aware approaches. So other groups have been converging on the idea of aging in the lung as a driver of COPD, and Dr. Peter Barnes in the UK has really been the champion of this concept of COPD as a disease of accelerated aging. And this is only a small set of the papers, but it really points to this idea of xenotherapy or aging pathway targets as a new horizon for COPD therapeutics. And this is a really important concept as we think of taking genomic medicine to precision medicine. I think the most convincing way to do that is to realize and internalize the idea that men and women may require different therapeutic responses if we're really going to use genomics and genomic insights to develop precision approaches to preventative and therapeutic approaches. So what does the next decade bring? The next decade is clearly one of promise. We look towards big genomic data, advances in machine learning, the development of polygenic risk scores. We know that better phenotyping of study subjects and subtyping using next generation analytics is really going to advance us even closer towards precision medicine. We have the opportunity to address sex and gender differences related to the progression of multiple domains or multiple phenotypes together with aging. We can think in terms of the development of subgroup-specific therapeutics, but we can also be visionary and think of ways to use our insights into the genome and the networks for prevention of lung disease, with careful considerations for prevention, starting with in utero susceptibilities. And this is very timely because we're very concerned about the continued emergence of sex and gender related differences in lung disease related to vaping and electronic cigarette use. And this is an area for further study. So as I think in terms of my own hope, I really hope we can bring together all of the elements we've discussed in this talk today, thinking in terms of development as an early stage of aging, thinking in terms of sex hormones, what happens at key transitions in life between menopause or the andropause in men, bringing together the layers from genetics, epigenetics, transcriptomics, proteomics, and metabolomics, and putting them together in a sex and gender-aware omic framework to really get to this eventuality for both biomarkers and therapeutics for precision pulmonary medicine. I'd like to thank you for your attention. I received funding for work in sex and gender differences in the lung disease from the National Institutes of Health, from the Brigham and Women's Hospital Connors Center. I have an Ignite Award for the development of sex-specific therapeutics, the Alpha One Foundation, and these are my institutional affiliations, and I thank you for your attention.